Okay, good morning, everybody. Happy Friday. Any questions before I get going with this? Last chapter. Okay, so this, uh, I'm going to keep it like this for just a second because I'm going to switch over to the textbook here in just 1 second. So I'm not going to make it full screen quite yet. This chapter wraps up the immunology unit of material, thinking about some potential issues, some uh, possibilities for our immune response to kind of attack ourselves, maybe, to go into overdrive, maybe, where it creates autoimmune situations that are ultimately big issues uh, on our systems. So there are numerous things we want to think about as we move through here, but a lot of it, again, is thinking about disorders or defects with the immune response that cause these adverse reactions. So what we've thought about up to this point really is when the response is functioning properly, when it's recognizing foreign invaders, mounting a response, and defending ourselves like we would want. But in these circumstances, something isn't functioning quite right. And it causes, again, some sort of type of adverse reaction within the body. And that's what we want to think about as we move through. Again, there are numerous things that we will kind of be thinking about as we progress along. So, initially, and like I said, I am going to toggle over to the textbook here in just a second. So, initially, we want to think about things like allergic reactions. There's all sorts of possibilities, and numerous people here would probably say you've got some sort of allergy. Hay fever, seasonal allergies. Right now, as we're starting to kind of warm up and the grass wants to start to grow, we can start seeing or thinking about some potential allergies to start plaguing us, which is probably a bit of a concern as we're, hoping to be moving out of all of this COVID stuff as we start getting some of these seasonal allergies. It kind of begs the question, what's really going on with us? Bee stings are something that are relatively common when we think about potential allergic reactions, and these have the possibility of being a serious issue. Potential for anaphylactic shock that we'll hit here in a second is a possibility with this. And some individuals that know or maybe ultimately find out as a result of a bee sting that they've got these serious allergies will find themselves oftentimes carrying an EpiPen or epinephrine if we're thinking some different brand name. It's a substance that will help to pull you out of this allergic reaction as we think about stimulating your blood pressure. So it counteracts this drop in blood pressure. It gives you, hopefully, gives you enough time to get your stuff together and get to the hospital, get to someplace that can treat you. And often you'll get other stuff, antihistamines and other substances to help deal with the situation. So I always am just thoroughly impressed when I see these images of different pollen grains and other allergens, but it's a lot of pollen that we're looking at here. And as we see these pollen grains under the microscope, this is an electron microscope that gives us these really, I think, kind of stunning images. You see some of these structures, these pollen grains here, and if we were to zoom in on one of these, the green or the yellow guys right here, you could see uh, the whole surface of these pollen grains are full of little spikes. And I feel like deep down, that's got to be something that's bad news if you're inhaling that. Lots of us, no issues. We come in contact with it and our body doesn't think anything of it. But there are others that do have allergies. And I'm like, well, it seems like a pretty good idea that you'd want to mount a response against this. These seem like some sort of crazy danger to our system. So they've been colorized to just kind of make them pop out, but they can trigger all sorts of allergic types of reactions, rhinitis, runny nose, hay fever, in individuals, again, that are sensitized to these specific allergens. And we'll talk about what's happening with the sensitization process here in just a second, all right? There's other possibilities when we think about allergic reactions, some skin reactions, rashes, maybe hives as a result of coming in contact with something. Oftentimes we might think about foods that can stimulate a rash. It could be different types of like laundry detergent or something like that, that maybe you've changed a brand and your body mounts a response against something like that, and you can see some of these reactions. Peanuts are another one that can provoke a pretty serious allergic reaction. Again, EpiPen worthy, possibly. All right. There's definitely differences as far as how severe these responses may or may not be. But I feel like you definitely, if you are in the situation where you can develop these severe types of responses, carrying something like that with you is something that you should uh, do. It's definitely needed. Let's see. I'm going to stop sharing this and move to the text real quick. Let's see. I can't remember. Yeah. Okay, wait, I've got to share it. Oh, so many things here. Okay. Sharing. Maybe, can I move this over? Okay, so before I get to that figure, there were a couple of tables in the text that I did want to touch on as we think about some of the mechanisms of the response in the first place. There's numerous cells and numerous substances that these cells potentially will unleash that stimulate some of these specifics that we'll be thinking about as we progress along. So there are numerous potentials, and this again, right back to some of these allergic or the allergens and the responses that you might see with that. So one mechanism that we will focus on in the next figure here in just 1 second is the interaction with this response, recognizing these specific substances, maybe allergens, as being bad news and mounting a response targeting mast cells, linking up with them and initiating what we're going to think about here in just a second. So when those mast cells get activated, get sensitized, if you will, they basically get told, we've seen these bad guys, here is a trigger or a receptor that will allow us to detect them, and when you see it, you can jump into action, all right? And so inside of these mast cells, there's handfuls of different types of granules, digestive enzymes, and substances that could stimulate other cells to respond. So a few of these granules play an important role when we start thinking allergic reactions. So heparin is one granule. It stimulates the generation of bradykinin that increases permeability, vascular permeability, increases vasodilation, bronchial constriction, so closing off of the airways, and it also stimulates excess or increased mucus secretion or mucus production. So runny nose as a possibility with that. Histamine stimulates smooth muscle contraction, more vascular permeability. So all of these, we're trying to help get immune cells into this situation. Maybe that's good. Maybe it's not good if we're thinking some of these, again, adverse situations. So more increase in mucus production and tear formation as well. Watery eyes, often a good symptom when we start thinking hay fever. Serotonin also increases the vascular permeability, more dilation and smooth muscle contraction. So they all act kind of in a similar function, but different molecules targeting or acting a little bit differently as we get through there. Again, trying to stimulate this type of a reaction. So some other mediators when we think about the allergic response. Leukotriene causes smooth muscle contraction, more mucal secretion, and vascular permeability, similar to what we were thinking about. Prostaglandins also with the smooth muscle contraction and vasodilation. And then tumor necrosis factor, this is a type of cytokine. It stimulates inflammation. It causes the production of more cytokines, which in turn target or stimulate other cells to come in and respond. And there are boatloads of different types of cytokines, chemical messengers that are utilized in an immune response, and dependent upon those messengers that are released out, dictates the types of cells and the types of responses that we might see. So this next figure, I'm going to keep it right in the textbook and just move through it because even when the PowerPoint is on full screen, it's not as easy to see as it is right here. So I'm going to keep it right like this. And we'll just kind of scroll up and down a little bit to talk about it. So initially, in this immune response, we've got our antigen presenting cell, so a macrophage as an example. It comes in contact with our foreign invader. We could come in contact with maybe one of those pollen grains as an example, all right? So we come in contact with this substance. In this case, our first exposure to this allergen, we would say this is the sensitizing dose. This is when we first recognize what's happening, all right? We're becoming sensitized. So we engulf it here. We're phagocytizing this antigen. It could be that pollen grain. We in turn will destroy it and get little fragments. Those epitopes get presented on the surface of that cell. And now we're going to call it, once we start presenting, once we do the show and tell, we call these guys antigen presenting cells at this point. So on their MHC2 molecule, it's linked up with that specific epitope. And as we described before, thinking normal response, there's potentially multiple epitopes that are being presented by these cells, right? And with that said, there are possibilities, but we're stimulating other types of an immune response, maybe a normal response to deal with this. And some of these fragments can potentially stimulate other stuff, all right? What we're thinking about right now, all right? So this is the issue. So this MHC2 molecule stimulates, in this instance, a T helper 2 cell. And recall the T helper 2s are important with humoral immunity, with antibody production, right? So they stimulate B cells, right? So the T helper 2 cell recognizes this specific epitope. It's CD4 molecule, they dock together, they exchange, and now you can see our T cell linking up to the B cell receptor, showing that B cell the specific epitope, right? In this instance, as the T cell snags that antigen, it basically does a little bit of recognition with it, if you will, and it starts secreting specific cytokines. And like I said, there's a boatload of different types of chemical messengers that might be stimulated to be produced, all right? So in this case, The T helper cell releases a couple of cytokines called interleukins, interleukin 4 and 13. Both of these guys activate, stimulate a B cell to show up, all right? And they also help these specific interleukins, flip it back up here, they help those B cells as they originate to be kind of told, if you will, because at this point, the B cell is not making antibody. It has to see that T cell's got to see what it's showing it. So these interleukins help to stimulate these B cells to basically start producing IgE, right? And it's this class of antibody that's important when we think about allergic reactions, right? It's also important with parasitic infections as well. We'll hit that here in a bit, all right? So it starts pumping out IgE, again, stimulating and the immunoglobulins, those antibodies that are going to be helpful in mounting this type of an allergic reaction. Maybe that's not best to call it helpful, but that's what they're going to do. All right. So these cytokines task these B cells and say, you know, what we ought to be making is IgE. All right. So they differentiate and turn into these plasma cells. The plasma cell, again, is basically an antibody pumping out factory. All right. So they just start producing mass quantities of IgE. All right. There's other stuff that, you know, if you've read through the text, which I hope you have, there is the potential for these B cells to go through what we call class switching. So you could have an antibody or a B cell make an antibody, and it can transition and shift over to producing a different type of immunoglobulin. Usually we see this happening with the B cells making IgM. And if you remember back when we were talking initial antibody production, IgM is the first one that gets produced. And then we start thinking about making IgG, all right? So maybe these cells start off normally functioning like they should, and they're producing IgM. But through the recognition of some of these interleukins, it may stimulate those that were making IgM to switch over and start making IgE. Now we've got the potential to mount an allergic reaction, all right? So again, this is not something that we would really think of as a normal situation, all right? The production of IgE. This is an adverse type of situation, right? Where these cells release these other messengers that are like, you know, what we ought to do is think about this other stuff, right? So those IgE producing plasma cells release all of this IgE. This immunoglobulin links up to mast cells. There are receptors on the mast cell for IgE molecules. They link up, and at this point, this mast cell has been sensitized. It's been told, when you see these bad guys, these specific antigens, when you see these guys, you're going to respond, all right? So at this point, typically, by the time this has happened, by the time we get to number 4 right here, that specific allergen, that substance that stimulated this, it's probably all gone. And you don't see a real reaction as a result of this first sensitization. If you do, it's pretty minor, all right? It's the next go around, number 5, the subsequent exposure. And this is something that we would call a shocking dose. This is when you see it the second time. This is when you actually experience that allergic reaction, right? So this next go around, when you encounter that allergen, those mast cells that have already been sensitized, they already have those immunoglobulins stuck on their surface. They can rapidly recognize those allergens, and the response is very quick, all right? So it causes those mast cells to degranulate, and all of those substances, the few that we mentioned, flipping it back up here, heparins and histamines and serotonins, amongst some other stuff, helps to stimulate inflammatory response, helps to bring in other cells with the vasodilation, You see watery eyes and mucus secretion with some of the symptoms that you might be exhibiting, all as a result of these mast cells, right? And for those of you that exhibit allergic reactions, you oftentimes take Benadryl or something like that, an antihistamine, something that blocks the production of histamines, or not the production, but the release of histamines from these mast cells. And that drives a wedge in this whole process, right? Okay. So let me see. I feel like one more table. And this does fit right with this first bit, thinking types of allergic reactions. What we've really just thought about are classified as the type 1 hypersensitivities. And type 1 hypersensitivity reactions rely on that mechanism that we just worked through, those histamines releasing these substances. So there's a couple of common names for these differences in severity for sure. There's allergy-induced asthma from specific allergens that are inhaled, and you can see common symptoms, constricted bronchi and trouble breathing. Anaphylaxis is definitely a severe, potentially fatal reaction. So it's a systemic reaction. So throughout the body, triggering all sorts of serious issues, hives, itching, swelling of the tongue and throat, potentially making it almost impossible to breathe, nausea, vomiting, low blood pressure, and potentially shock as a result of that. Hay fever, pollens or molds that are inhaled. This is a very mild situation, runny nose, watery eyes, and sneezing. And then hives, skin manifestations, food or drug reactions, oftentimes, or maybe some insect stings can fit that as well. Dependent, it could also potentially bump you up here to anaphylaxis. So raised bumps, potentially kind of all coalescing and causing some serious issues, right? So there is really some interesting stuff thinking about some of these things and some of the IgE in the first place. We've mentioned this a little bit before, and the text talks about it in some detail for some of these potentials are maybe the result of what we think of as a hygiene hypothesis. We're too stinking clean is the problem. Is that a problem? Maybe, maybe not, all right? Oftentimes, if you think about someplace else, maybe a third world country, the potential for all sorts of parasitic infections are pretty tremendous, all right? It's likely, as a child that you're going to contract some sort of a parasite and maybe handfuls of different types of parasites, right? Worms, they're not unheard of in the United States, but they're pretty uncommon, all right? But elsewhere, maybe not so uncommon. And IgE molecules and these types of reactions that we're thinking about right here can become super important if you're mounting a response or a defense against some of these massive worms, right? And it truly helps to protect us, right? But then you think, again, this hygiene hypothesis, what happens to your body that wants to constantly be doing this protection when there's nothing that it needs to protect us from? Maybe there's other potential issues that we might see, right? And stuff like some of these hypersensitivities, some of these allergic reactions, some autoimmune diseases, as our body kind of, is idle with this immune response, has the capability of targeting other things. And that's a big issue, right? So it kind of begs the question, could there be a potential to having some parasites and alleviating some of these other issues, right? And that is a story in and of itself. And I'm not going to go into great details with that. But there definitely are some potentials. And you can't just readily go to the doctor in the United States and ask to be prescribed some worms. They kind of frown on that. But there are people that grow them in their own bodies, and they will harvest them and sell them to you. If you are truly interested, you can search up some hookworm therapy, and you will see some potentials that may or may not help you. All right. I think if you've got hay fever, probably getting yourself some worms is not the best idea. All right. But if you've got other serious issues, maybe that's not so bad. So kind of crazy. So this right here is the hygiene hypothesis that talks a little bit about some of that stuff, which I think is pretty impressive. And yeah, there are definitely some possibilities with that. So that's all. Type 1 hypersensitivities, different types of allergic reactions from mild to severe, thinking, common allergy to the anaphylactic shock. The next one's the type 2. These are cytotoxic hypersensitivities. I'm just going to keep it on this again for 1 second so we can move through. A lot of it, we think about blood transfusions. So the ABO blood grouping system that we'll see here in a second becomes super important, where we may or may not recognize specific cells as an issue, all right? But there's other issues as well. And we can think about hemolytic disease of the newborn. This is the Rh factor, and we will talk about that here in just a second, but it can cause some serious issues in that unborn child. And then transfusion reactions, so you get the wrong blood type. These are both types of type 2 hypersensitivities, all right? And with that, there's definitely some issues. Let's see. Let me scroll down here and see what's up, if there's another table. Okay, I'm going to stop sharing this and go back. There's my PowerPoint, go back to this. Okay. So this is something, the blood type. This is something you guys might know what your type is. Maybe you don't. Maybe you will find out. I think maybe you do blood typing in anatomy and physiology lab. Maybe not. All right. I have heard all sorts of issues with that, and I hope that that's no longer happening. But somebody told me, this was years back, but they were doing blood typing. You've got these little lancets, these little needles that will poke your finger and make it bleed just the littlest bit. You're poking yourself. You're getting your own blood. You're putting it on the card. You're doing the test. That seems all fine and good, right? I was teaching an immunology lab. I wanted to do this stuff. They're like, oh, no, you don't do that. We can't do that. That's blood, right? That doesn't make sense. I hear that the A&P lab is doing it. I'm like, well, that doesn't make sense. How can they do it and I can't? Then I hear some other stories. And in the A&P lab, again, this was back a few years back, they were reusing the lancets. So you'd get a group of students and everybody used the same lancet and poked their finger. I'm like, what the hell are you guys doing? That's an issue, all right? So you may test your blood. You may do blood typing. Maybe you won't. But if you do, make sure you're using your own lancet, a clean one, right? A sterile one, and you're not sharing it with your neighbor, right? That's a serious issue. All right. So the blood types and the blood grouping system, there are 4 blood types. There are multiple ways that you may potentially get this. Quick a quick and dirty genetics. You've got two chromosomes, two alleles that potentially can generate your respective type. We're only showing you their blood type. So in this instance, on your chromosomes, you may have two A's, or you may have one A and one O chromosome. The O is recessive to the A's and B's, all right? So two A's or an A and an O, you're type A. Two B's or a B and an O, you're type B. One A allele, one B allele, you've got both. This is codominance, and both of these alleles are expressed equally. You're type AB. And so you know, genotypically, you know, these are your specific alleles, 1A, 1B. A type O person also knows their specific alleles because they have two O alleles, right? There's nothing else that they could have, all right? So AB person and an O person knows. The A or the B person technically doesn't know what their genotype is. They could reproduce and potentially know based off of what their offspring are. Maybe they know, and maybe they don't know. Just depends on what their blood types possibly are, all right? So knowing what your type is important. Also knowing if you are a positive or a negative, which I think is the next slide, or maybe it's the second slide down here. That's also important. And it becomes important when we think about potentials with hemolytic disease of the newborn. So again, we'll hit that here in just one second. So we can see in the top of this figure, our respective red cells. And on some of them, there are glycoproteins. So these are sugar protein combos. And these glycoproteins are antigenic, right? We can recognize these things, all right? So the type A person has A antigens on the surface. The B person has B antigens, and the AB's got both of those antigens, right, on their surface. The O person doesn't have any antigens on their surface, right? No antigens. Circulating with those cells in the plasma are where we find immunoglobulins. And there are all sorts of antibodies coursing through your body all of the time, all right, to help to keep us protected, all right? And we've got, dependent upon your blood type, dependent upon your blood type, we've possibly got antibodies circulating around. So if you have type A blood, you've got B antibodies. We call them anti-B antibodies circulating. Type B's got anti-A. A type AB with both antigens on their surface, they don't produce any antibodies, or they wouldn't be alive, all right? There'd be issues, all right? So there's no antibodies produced. And the O person with no antigens produces antibodies against both A and B, all right? This should start to make sense as far as who could give blood to whom, all right? Based off of what sorts of antibodies you may or may not have in your circulation, or what sorts of antigens you may or may not have on your erythrocytes, all right? So type A person can donate to a type A person, all right? Or to a type AB. The B can donate to themselves, the B groups, or to an AB, all right? And again, because there's no antibodies with these guys. AB people technically can receive blood from anybody, all right? Because they don't have any antibodies, all right? So they're what we call a universal receiver. They can get blood from anybody, but they can only donate to similar AB people, all right? Because they've got both antigens on their surface. So the O person is not having any antigens on the surface is what we call a universal donor. They can give blood to anybody because there's no antigens, but the O person can only receive blood from other O people because they've got antibodies against both A and B, right? So Red Cross really likes the O people, right? Because their blood can be given to everybody. They also need O blood for O people that need blood, all right? That's definitely something that's needed, all right? So with that, this is just the straight up blood grouping, all right? There are the positives and negatives that we need to throw this into the mix as well. This is the RH factor. I think this is my next, well, we'll do this first. Then we'll talk RH. So here we've got, with the donation, we've got a type A donor with their antigens. We've got a type B person that's going to get this A blood, all right? So as the donor gives this blood to this recipient within their circulation, as I said, they've got antibodies against these other types of cells. Maybe they do. So the B person does, and they've got immunoglobulin against A. When they are given A blood, as we see in this next frame, that A blood is quickly recognized by these immunoglobulins, and it's snagged, making an agglutination reaction or hemagglutination, talking red cells, and you get a clot that forms as a result of this. Dependent upon how much blood was transfused, sets the stage for possible fatal transfusion reaction, as you see massive clotting occurring in the patient, right? Kind of depends. As this happens, IgM is one of the immunoglobulins. If you recall, IgM and IgG both can stimulate complement proteins. So as the IgM molecule links up these guys, it stimulates complement proteins, and complement causes these red cells, the bad cells, to be lysed, to be destroyed, all right? Hopefully, getting rid of these cells before the blood clot that's being formed becomes a serious consequence, all right? So this is a transfusion reaction, all right? Hemolytic transfusion reaction, the wrong blood type, all right? Giving A blood to a B person, all right? Bad news, all right? The RH stuff, on the surfaces of cells, there's the potential for all sorts of other antigens, more than just what we showed with the simple ABO blood grouping, all right? And RH is one that is something that's really pretty reactive. There are some other antigens that are a little less, but there are definitely some potentials, all right? And dependent upon your specific genetics, there are maybe issues where you will have adverse reactions, and you need to know some of the specific finer details about your blood groupings to help provide some protection, right? We're not going into all of that stuff right now, right? But the RH we do want to think about, all right? So this is something that's just simple Mendelian genetics, all right? You can have 2 copies of the protein or of the gene, 2 copies of it, and you are positive. You can be heterozygous, 1 copy and 1 missing, or negative, if you will, and you are still a positive. To be negative, both of those genes lack that. So if you're negative, what your genotype is, right? You are lacking both of those alleles. If you are positive, well, it's a 50-50 chance. You are either homozygous or heterozygous for that specific allele, all right? And maybe having offspring clues you into that if their blood type is different, right? That's the issue, all right? So deep down, really the issue is with the mother, all right? And if the mother is negative and the child happens to be positive, so if the father is a positive, there is a chance that that child will be positive, all right? And again, genetics, it depends. If that father is homozygous, 2 positive copies, if you will, every child that he produces has that positive allele, so they'll all be positive. If he's heterozygous, well, then it's a 50-50 chance. And you could have a negative kid or you could have a positive kid, all right? It just depends, all right? Back in the day, love is in the air. You decide maybe you and your sweetheart think you ought to get married. Not that should be, what prevents you from pumping out a kid or not. You don't need to be married for that to happen. But back in the day, if you wanted to marry someone, they wanted to do a blood test to see if you're compatible. And really what they want to do is figure out what sorts of likelihood for this to occur. Could you generate hemolytic disease of the newborn? If you could, you probably don't want that, right? I'm pretty stinking sure, right? So they would advise the prospective couple that maybe there's some other fish out in the sea for you. Maybe you guys shouldn't get married. And I step back and I wonder, if you've got two people that are smitten, if they're in love, if they want to get married, do you think a blood test is going to prevent them from getting married? Probably not. Maybe. I mean, if they're not going to sign the marriage license, that's pretty good to prevent you, right? But there's issues, right? We now have ways around this. And there are some treatments that will prevent this from happening in the first place. Lots of places no longer require blood tests if you're looking to get married, right? And like I said, not that marriage is going to, one way or the other, allow you to or not to get pregnant. So the issue, you've got a negative mother and you've got, in this case, a positive child. This child, it is highly likely that as this child develops, and typically it's by the time we make it into the third trimester or right as they're ready to hatch out, as these children are being born or right at the kind of tail end of the pregnancy, there's the possibility of some fetal and maternal blood to mix. Definitely on the potential for delivery, there's the possibility of some of that. And now it's just like the mom has been vaccinated or immunized with this positive blood. She produces antibodies against that positive blood and recognizes it as being foreign. For the first kid, it doesn't matter. This is the sensitizing dose. Nothing happens to that first kid, all right? It's the next go around. If that second child is also a positive, her body recognizes that as being foreign, as being an issue, some sort of possible invader, and now what we need to do is destroy it. IgG is the immunoglobulin that is important. It crosses the placenta. It provides protective immunity to that child. As it hatches out into the world, it gives it kind of its first bit of help, if you will. It also can cause serious issues. In this case, it recognizes the fetus being positive, crosses the placenta, targets, and tries to destroy it from within. Maybe it does. Spontaneous abortion is definitely a potential for this. Maybe that's not called spontaneous. I think it is. So if it is born, it has some serious issues. There are possibilities to correct this. That child requires immediate blood transfusions to basically, there's all sorts of immune complexes where the IgG is linking up to the red cells. So basically removing all of the blood and replacing it and maybe doing it twice helps to rinse all of that away, and you can save this child's life. You can see some super nasty images of hemolytic disease of the newborn, disturbing to say the least. So like I said, we do have ways around this. There's a drug called Rogam that basically blocks the production of these antibodies. So the mother can be given this drug. Usually it's 2 shots. They give it halfway into the pregnancy, and then right before labor and delivery occurs another dose of it. This prevents the antibodies from being produced in the first place. So basically, it keeps the negative mother from getting sensitized with these respective deliveries. So each time, basically making it like it's the first time and not setting the stage for serious issues for the destruction of that fetus. So it's important to recognize not only what your blood type is, but are you positive or negative, all right? And then with that said, females, if you are negative, there should be at least a little hint of that in the back of your mind. What is this child or the father of that baby? Maybe you know, maybe you don't, right? It makes me kind of curious about other issues, potential, maybe a miscarriage or, something earlier in life that maybe goes unrecognized as far as a positive, negative sort of situation. And maybe that woman becomes sensitized unknowingly, right? Sensitized. So no ROGAM was ever administered. And then any subsequent pregnancy that is a positive child, we would see this resulting. And so potentials with infertility, numerous miscarriages and things like that could be the fact that maybe that mother had been sensitized unknowingly against that positive blood. Kind of crazy to think about. All right, I think with that I will stop. That's a good point because we will be picking up with the other stuff. We'll hit that on Monday. Any questions or concerns? Okay. Thank you all.